In an earlier video, I showed how to make a sealed microecosystem that can keep microbes alive and interesting for several weeks. I've made a couple dozen of these over the past few months and observed and recorded lots of cool stuff that I think is worth sharing. I'm getting quite a variety of results. Slides start out with different kinds and populations. Some species proliferate over days while others go extinct. It's hard to know how best to organize and present results, but for this video, I'm going to focus on just one slide, prepared December 22nd and labeled 1222A. I added algae and water from a culture derived from pond water to a weighed slide, checked for activity, added a trace of goldfish food, weighed again, placed the Vaseline-treated slip and weighed again. This shows the whole slide overview when it was first made, then again a week later. It starts in real time, then goes to time-lapse to better show microbial motion. 25x speed, 100x speed, 1500x speed. The worm-like things are stenostomums and an aleosoma, and the smaller things are mostly rotifers and Paramecium bursaria. The white line by the goldfish food crumb at the lower left moves over time, showing where the bacteria are happiest. The blue rectangle shows the area viewed in the next portion. Here, using a 4x objective, you can see the goldfish food crumb surrounded by several Paramecium bursaria, the aleosoma, and a couple of the stenostomums at normal speed, then 25x, and 1500x. The following are close-ups of several of the species on the slide. You can see them in 3D using red-blue glasses. First is the aleosoma using a 10x objective. I find that 3D viewing is especially helpful in making sense of images with a complicated algae background. Next, a stenosomum, also with 10x objective. Next, another stenostomum, this one soon to split into two. You can see the new mouth at the midpoint. Next, a loxophyllum. And a stentor. Now, with a 20x objective, a coleps and Lepidella rotifer. And a haltotier. and a Paramecium bursaria feeding on bacteria with 10x, (20x), and 40x objectives. And an amoeba and colonial algae, Eudorina, with 20x. And a testate amoeba with 10x. This shows the notes I took on preparation and observations of the slide. Note all the microbe species I observed on the first day. For each day of observation, I measure the weight of the slide to check for evaporation, then spend 5 to 10 minutes scanning the slide with a 10x objective using a set of polarization filters to view in 3D. As you can see, I ran out of space on the page and squeezed in species at the edges, notably the Aleosoma and Stenostomums. My note-taking on these slides is a work in progress. At 16 days, some of the species are increasing, notably Paramecium bursaria and haltoteres. Lepidella rotifers are holding steady at 5 or 6. The single Aleosoma, nematode, and bedieloid rotifer are still there. The Stenostomums are starting to decrease. The Stentors, Amphileptus, and Loxophyllums have all decreased to 0 observations. Other slides show different progressions with coleps or gastrotrich prevailing over other species. I plan to make a couple more videos focusing on single slides, then maybe a compilation of interesting observations. If you've watched this video to the end, thank you. I've included more information and links in the description. Questions and comments are welcome.